Hello guys, welcome to my class, and today we will discuss or take up a new chapter, new topic, that is immunoassay. What is immunoassay? Immunoassay is a technique of qualitative and quantitative estimation of immunoglobulin and antigens. ठीक है? Qualitative and quantitative analysis mean के qualitative stands for qualitative means के what type of molecule is present in a sample, that is the qualitative test. If that particular compound is present or not in that sample, that is also qualitative test. And that means what type of substance is present in that sample is qualitative test, and how much amount of that particular molecule is present in that sample is known as quantitative analysis. Okay, so this is used for qualitative and quantitative estimation of antigen and antibodies. That means that these two are antigen and antibodies are associated with immunity, and we are assaying, we are analyzing qualitative and quantitatively the component associated with immunity. Then this assay is known as immunoassay. Okay, so this is the qualitative and quantitative analysis of antigen or antibodies, and this is based upon the interaction between antigen and antibodies. Whenever there is a antigen met its specific antibody, they will bond with each other, forming the antigen antibody complex, that is known as interaction. They bind each other to each other at their specific points, like paratope on the antibody and epitope on the antigen. So this is the point which used for their binding leads to the formation of antigen antibody complex. So this assay is based upon their interaction or binding with to each other. Okay, so there are two types of technique used in the immunoassay. One is radioimmunoassay, that is the RIA. Radioimmunoassay means के a radioactive substance or element or compound atom is used in this technique. Okay, radioactive isotope of any compound like nitrogen 15 in iodine. Okay, radioisotopes of iodine like this. Okay, used if that radioactive substance is used for the determination qualitative or quantitative analysis, then it is known as radioimmunoassay. If in the technique any enzyme is used which is tagged, we will discuss later. If enzyme is used as a marker, okay, we can say it as a marker. If enzyme is present in place of a radioisotope, then it is known as enzyme-linked immunosorbent assay or ELISA. Okay, so these are the two types of immunoassay techniques. Okay, so today we will discuss about the RIA. The RIA is specifically used to determine the presence both qualitatively and quantitatively antigen in a sample. Okay, presence of an antigen in a sample, both in terms of both qualitative and quantitative. That means what type of antigen is present or how much that antigen is present in that sample. This is frequently used in diagnostic laboratories or research laboratories. Okay, so this RIA is used for the determination of antigen in a sample. Okay, and this RIA technique is based upon the competitive binding of labeled and unlabeled antigen. If you take antigens and separate it into two groups, one group is made labeled, or the one group is attached to the molecules of one group are attached to a radioisotope, then this antigen, group of antigen of same group, these antigens of this group will known as labeled antigen, and the second group of the same antigen which are not attached to any radioisotope is known as unlabeled antigen. If these two antigens are added to their specific antibodies, both these groups and both the antigens of both these groups will compete with each other for binding to that antibodies. That means both of them will try to bind itself to their specific antibody. That means they will compete with each other. They will compete with each other for binding to their specific antibodies. So this is the basis of the analysis. Okay, we will understand this by this diagram. Suppose, first of all, we take the antibodies, specific antibodies of those antigen which has to be analyzed in the sample. Okay, we take the specific antibodies for this and that antigen, okay, which is to be analyzed, and this antigen is incubated on a glass surface, and this antigen will bind to this glass surface. Suppose this is the glass surface and these are the antibodies attached to this surface. And in the next step, the antigen of two types, two versions of same antigen is added to this test tube containing the bound antibodies. Both these antigen are of same nature, is specific to these antibodies, but one is labeled. That means that these antigen are attached to an isotope. Suppose the label it is okay. This is the iodine isotope. Suppose anyway this is attached to a radioisotope. It is radioisotope. Okay, so that means this is the labeled antigen. If whenever this antigen will present, these isotope will radiate radiations. If this radiation is estimated by using any radioactive counter like gamma counter, we can estimate the radiation, how much radiation is present, okay, and by this radiation we can determine the amount of these antigen to whom this radioisotope is attached. That means by the radiation of this attached radioisotope we can determine the amount of these antigens. Okay, so suppose this is the radioisotope, okay, labeled antigen, okay, and some of the unlabeled antigen of same nature. Okay? Here we can see that the amount of labeled antigen is more than the amount of unlabeled antigen. When these two are present in the test tube along with their antibody, they will compete with each other for binding to their specific antibody. Since in this figure the amount of labeled antigen is more than the amount of unlabeled antigen. That means के chances is there कि these more labeled antigen will bind to this antibodies, and the amount of unlabeled antigen which bind with this antibody will be less. Okay, because the amount concentration of labeled antigen is more and the unlabeled antigen is less, their chances will be less to bind with antibody. So suppose this happens, so here we can say that K out of 5, 4 labeled antigen bind with these antibodies, and out of 3, 2 bind with their specific antigen. This is the competition. By then in the next step we wash the whole test tube, and the unbound antibody will be discarded, washed off, and only antibody which are left in the test tube will be bound antibodies. Okay, so we cannot estimate this, but we can estimate the labeled antigen using a radioactive counter, radioactive like gamma counter, radioactive counter like gamma counter. With the help of gamma counter, we can count the radiation coming out from this radioactive isotopes attached to this antigens. Okay, by estimation of their radiation, we can calculate the amount of antigen which are labeled with and attached to these antibodies. Suppose कर लो कि these labeled antigen which are bound with their specific antibodies, their concentration is A. In the next experiment, suppose we take equal amount of both labeled and unlabeled antigen, and the amount of antibodies is fixed. Amount of antibodies is fixed, but here the amount of radioactive antibody is also fixed, but the amount of unlabeled antigens, amount of labeled antigen is fixed, but amount of unlabeled antigen is increased over here. That means here the concentration of both labeled antigen and unlabeled antigen are equal. Suppose this is, you can see from the diagram that it is the labeled antigen has 5 molecules and unlabeled also is 5 molecule concentration. Okay? They will compete with each other because they have equal concentration. They will compete equally with each other for the binding to the antibodies. That means in the next step, the chances that out of the 5 of both the type, 3 of each type of antigen will bind to the antibody. There are specific antibodies. Now they will radiate the radiation, and the counting will be done of their radiation, and by this counting we can estimate the concentration of this antigen. Okay, labeled antigen, suppose here the counting is B. Here the counting is B, that means the concentration is B. Because suppose here there are 4 molecules are bound. Okay, here only 3 molecules are bound. That means कि radiation by these three bound antigens will be less than the radiation which is coming out from the four bound antigen. Okay, है ना? So this radiation will be more than this radiation. Okay, by counting this we can calculate that the amount of these antigen which is B. That means by calculation we can say A will be greater than B. Okay, how? That means concentration of bound labeled antigen will decrease as the amount of unlabeled antigen increases. That means amount of concentration of unlabeled means that those antigen which will be present in the sample. Okay, concentration of labeled antigen if increases, the concentration of bound labeled antigen will decrease. Okay, है ना? So by this method we can calculate the quantitative, we can calculate the concentration of unlabeled antigen present in a sample. By this will be done by a graph. In the graph we plot a graph by using constant concentration of labeled antigen. Okay, here we also have constant concentration of labeled antigen, but the unlabeled antigen, concentration of unlabeled antigen is increasing. So we take constant concentration of labeled antigen and variable concentration of or increasing concentration of unlabeled antigen. Okay, so suppose we take various concentration of unlabeled antigen and each step we calculate the amount of bound labeled antigen. So as the concentration increases of unlabeled antigen, the concentration of labeled bound antigen will decrease. Okay, at each step, at each concentration increasing, when the concentration is of unlabeled antigen is gradually increased, the concentration of bound labeled antigen will decrease. If we plot a graph for that, and on x-axis we take the concentration of unlabeled antigen which is gradually increasing. On the another y-axis we take the concentration of bound labeled antigen through the by estimation of this counting of the radiation using the gamma counter. Okay, so we will get a graph like this. Okay, so suppose in the unknown solution we take the known concentration of antigen and known concentration is of antibodies, but the sample along with that the sample is also added. The sample contains the unknown concentration of that particular antigen. Suppose we mix both of them we get a result of bound labeled antigen which is equal to this point. Okay, suppose this is the A. Suppose this is the A. By plotting, by the use of this graph, we can calculate the amount of antigen which are present in the sample. So this value the result we get will give the amount of unbound antigen present in that sample. So by in this step, we first take the known concentration of antigen, unknown concentration of antigen. That means that the antigen present in the sample, and this is the bound antigen surface which is already prepared. We add both of them. Okay, mixed with each other, they will bind. The whole solution will be kept for some time. Okay, here we can say that. You take test tube. These are the bound antigen. Put the radio unlabeled as well as the labeled antigen here. This is fixed amount. This is fixed amount. Suppose now this is also the sample. Okay, this is the sample containing, suppose this is the amount of unlabeled antigen. When they will put here in the next step, they will be incubated at a particular temperature for some time. Incubated, for some time. In the result, then what we will get? We will get some of the antigen. Suppose we took only 3. Suppose, okay. Okay, then we will wash it. Wash means all the unbound antigen will be washed off. So we will get the same result because we are taking very small amount as a symbolic way. Okay, we will get like this. We will get like this. Anyway, this time, okay. Okay, now we will use these bound antigen for their counting of their emission. Okay, through gamma counter. By gamma counter will be emission will be counted, and by the amount by the amount of radioactive emission, we can calculate the concentration of bound antigen. Concentration of bound antigen. If this bound and concentration of this bound antigen comes on the y-axis at this point, this is the amount. We can calculate the amount of unlabeled antigen present in the sample. So this is the way of calculating amount of unknown antigen, unlabeled unknown antigen in a sample. Unknown means quantity is not known. We are analyzing the presence of a particular type of antigen in the sample, both qualitative and quantitative analysis. Okay. So suppose this is present here, we get the result here of unknown antigen. Of amount of unlabeled antigen. is the basic technique used in RIA. Hope you would have understood the process. Thank you very much.